Sarah Hemmel. Welcome to Dummerston. Sarah is no stranger to this neck of the woods. She did her undergraduate work at Dartmouth, being attracted back to New England because of family roots here, even though she grew up way down south in Oklahoma. After Dartmouth, however, she went back to Oklahoma for a master's degree, is that correct? Mm-hmm. But then decided to see what the West Coast was like, Northwest, and did her PhD up in Pullman, Washington, and later postdoctoral work at
Temple University.
Temple, that's right, Philadelphia. And so she was gradually working her way back to New England when UMass found out about her talents and decided, well, we need her to come, and awarded her the Darwin Fellowship. Now, is this Darwin any relation to the Charles Darwin?
It is, indeed.
All right. But this is by name of the fellowship only. Mm-hmm. Right. So without any further ado, Dr. Sarah Hemmel, and I'll let her introduce herself.
Well, thank you very much, Bill, and thank all of you for, again, braving the weather to come out and see this and learn a little bit about conservation genetics and genomics and how I use those tools to understand habitat disturbance of plants and animals. Yeah, as Bill said, and some of these pictures may be a little hard to see, but they'll be better on the video. I'm the Darwin Fellow at UMass Amherst, which is a postdoctoral position with the Organismic and Evolutionary Biology graduate program. So these are students who are studying ecology, evolution, anthropology, environmental conservation, and I get to be their mentor as well as their teacher. I also teach undergraduate classes and then do my own research. So as a conservation geneticist, you know, what really got me into this field is my love of going outside, you know, being out in the woods or in a field or in a stream, catching animals, collecting plants, and you know, that's the love of conservation and of biodiversity. However, I do spend a decent amount of time in the lab where I take those samples that I got from the wild and get the DNA out of them, target the parts of the DNA that I'm really interested in, and then get the data that I need to try and understand how we can better conserve these organisms. So it's a little hard to see, but this is my laptop. So most of what I do, like many academics, is sit on my computer. I do statistical analyses, I write papers and grants, and you know, all of those other things. So it's really, it's a great field to be in because I get to do these different things. I still get to go outside when I want to, but you know, I can be warm and indoors when I want to as well. All right, so to get into conservation genetics, I'll just start by talking about major threats to biodiversity that we're interested in studying and trying to fix. So these are not all of the threats, but they're some of the really big ones worldwide. Climate change, obviously, is a big threat to biodiversity. It's, you know, has the potential to destroy glacial habitats for animals like polar bears, coastal habitats for, you know, animals that rely on those areas, as well as people who rely on those areas, as well as actually taking, you know, the species range of a lot of species and moving where they're really suited to live outside of their range, or shifting it, you know, farther north here in North America as the temperature gets warmer, up in elevation as the temperatures get warmer, or evolve to those changes, adapt to them. Pollution is another major conservation threat, and you know, there's obvious effects on water, so you know, pollutants can have strong effects on amphibians and you know, any kind of downstream organisms, and this can be from agricultural runoff, mining, you know, industrial activities, etc. Over-harvesting of things like fisheries is another major threat. And you can't really see it here, but these are all fish that are being kind of netted in. And you'll hear more about fish and conservation with Lisa Komorowski's talk in next week, actually. Invasive species, they're more and more, becoming prevalent as a major threat because they can take over areas where there's really no natural predators or competitors for them. For example, this is a cane toad. So is anyone here familiar with the story of the cane toads in Australia? So some are, some aren't. So cane toads are native to Central and South America. They eat cane beetles, which the beetles eat sugarcane. So in Australia, they wanted to try and bring in a predator to get rid of these beetles that were destroying their sugarcane crops. And what they decided on was the cane toad, which is this huge toad. They're just massive. And they can, reproduce really quickly. They only, like most toads, only need a little bit of water for their eggs to be laid in and their tadpoles to develop in. And then they just develop into these huge guys that can just move really quickly and take over new habitats. So, people are studying what we can do to stop them and how the environment itself is actually restricting their expansion of their range in Australia. And then the threat that I'm really going to focus on today is habitat fragmentation and destruction. So what I mean by fragmentation is just the idea that if you imagine this was all forested at one time, and now it's been divided into, this little rectangle, this plot, this larger area, that's fragmentation. So it's fragmented, any animals or plants or other species that live in this forest probably aren't going to be able to survive living, out there in the part that's outside of their habitat. So that poses a lot of problems and challenges for us to investigate. All right, so this is the part where I have to get into a few definitions, and please ask questions if you're confused about anything as I explain it. Conservation genetics is a multidisciplinary field, and it's really a combination of conservation biology. And, there's a lot of different definitions of conservation biology, but I like to think of it as, the study and prevention of the loss of biodiversity. So, as conservation biologists, we are interested in, how the processes work that lead to, species or population declines, and what we can do to stop them and help, management of these populations. So it's a combination of this field and the field of genetics. And I'm just going to give a brief overview of what I'm talking about with genetics here. We're not going to go into any serious details. Happy to talk about the details, afterward. But genetics is really the study of heredity and how characteristics are passed on from one generation to the next. So, your genes, your genetic code contributes to the offspring, to your kid's genetics code, but it can change, between generations in little minute amounts. So genetics is really, a field that's been around several decades, and, generally we're focusing on a single gene or a few genes, and I'll get to what I mean by that in a second. Now, who here has heard anything about genomics? Does anybody want to try to define it for us? No
Study of the genome.
Study of the genome. Yeah, perfect. So, yeah, you got it. You get a prize. So it's the study of the genome, and when we go from genetics to genomics, it's really about scale. So instead of looking at a small part of the genome in a single gene or a few places in the genome, we're trying to understand the whole genome, ideally the entire thing, or sample from across the entire thing. So, what is a genome? It's essentially all of the genetic information that an individual has. So, any single plant or animal or person, or fungus would have their own genome made up of chromosomes. So you hear about humans having 23 chromosomes. Different organisms have different numbers. And that doesn't really matter for us here today, but just think about it as, this is your whole genome in chromosomes, but we can kind of, stretch it out in a long, row and actually look at the genetic code in the type of work we do. So if we go from looking at the whole genome to a single chromosome, then we can narrow down even further to where the genes are and areas that don't have genes. Not all of your genome is actually in use of, changing the way you develop or behave or, go about your day. There are parts of the genome that, aren't really doing anything as far as we know. But those parts are actually really helpful to us because they can tell us a lot about the history and the differences between different individuals and different populations. And another way to think about this is just that your genome is a book, then your chromosomes would each be a chapter in that book, and then the sentences in the book are each genes or parts of the genome that aren't genes, maybe wouldn't form a cohesive sentence. And just to illustrate that further, if we're looking at a single gene, there's 4 different letters in the genetic code: A's, T's, G's, and C's. And those will make up the sentence or the gene that'll actually code for some trait or some, expression of some gene, in your body. So, say a trait for eye color or, traits that turn on hormones to make you develop. So a single change in the DNA might change the way a gene works. So just like in the sentence, changing the H in hall to the B in ball changes this sentence. The gray cat ran down the hall versus ran down the ball. However, it doesn't always change the way a gene works, because you could have a change that's not within a gene and it won't have any effect on the health of that organism or its ability to reproduce and pass on its information to the next generation. So, any questions about this? This is as deep as I'm going to go into it. So, this is just a picture of an older sequencer, from like the early 2000s that can sequence your DNA, but it can only sequence little amounts at a time. It's slow, it's kind of expensive, and that's really where we were for a long time. Advances, in next generation sequencing have allowed us to use machines like this to actually sequence millions of fragments of the genome at once, and it makes it much more cost effective, and we get a lot more information. So that's really where things have shifted, and this is getting cheaper and cheaper to do. It used to really be a barrier in conservation genetics too, that the difference between doing genetics on a few parts of the genome versus looking across the whole genome was too much of a cost when you're dealing with limited resources. But now we're kind of getting past that hurdle. And they even have, this is actually a DNA sequencer. So they even have these little tiny machines now that you can drop some DNA on them, plug it into your computer, and get some genetic data from it. And yeah, I just saw this for the first time recently. It's amazing to me. It's really cool. So now we're getting to where people can actually go out and collect a leaf and immediately look at its genome, right out there in the field. So it's really neat. And along with this, the cost per genome has just decreased precipitously. This is for a human genome. So keep in mind that the human genome was the first really, really big project where we wanted to sequence the entire genome and then feel really good about the confidence we have in that sequence. So 2001 was when the first draft of the genome was developed. And if you can't read that, it says $100 million for that first genome. However, by 2006, when they developed the first really solid genome that we can now compare a lot of other genetic data to, the cost had gone down to about 10 million and then just dropped off the cliff to where today, a single human genome could be sequenced for about $1,000, which is remarkable. Keep in mind, not all species have these kinds of resources. We don't have a full genome to compare our data to for a lot of species, especially the species I work on. But we're getting there. It's getting more possible. And you may be familiar with genomics in your own lives. I mean, people have been using genetic tests for years to screen for diseases that might be inherited, or to try and do other things, or identify a disease that someone might have. But today you can actually get bits of your genome sequenced by just ordering this kit, paying $200 and putting a little cheek swab in your mouth to get some cells. You just send it off. Yes, go ahead.
Could you tell us, when you use the word sequence, what does that actually mean is done?
So sequence is essentially reading the genetic code. So when I talk about the original sequencers that I first showed, what you get back is actually just a screen full of A's, T's, G's, and C's. And you can compare those across different individuals. And so there's just databases of these sequences too out there. Does that answer your question?
Yes.
What is the price? What's the cost? Is it the database? Is it to run the computers? Is it to hire the scientists?
All of the above, yeah. So the biggest cost right now for a lot of conservation genomics work is the actual sequencing process, because to really do anything that you feel confident in, you end up sequencing parts of the genome many, many times. And you need that duplication to really feel sure that you got the right part of the genome that you were looking for. So that can be quite costly. Generally $1,000 to $3,000 for up to 96 individuals at a time. You could maybe do 2 or 3 times that, but get much less information for each. And then there are some costs with the preparation before, extracting the DNA is relatively inexpensive. There are a lot of different protocols out there. People have been doing it for a long time. You can use things like chloroform, which is always fun in the lab to help extract DNA. And then, actually getting from the DNA to the product that you're going to send off to a lab for sequencing, that's a little bit expensive too. Yeah. When you think about time, so a lot of the tools that we used, that we've been using before genomics actually take up way more time. So when you think about how much someone's time is worth, it really balances out, and now it's shifting much more toward doing genomics.
Yeah, you just send it off to a lab. For example, in Amherst, UMass, do you have sequencers on campus?
Yes, we have a genome resource lab in the biology department, and they have a couple different types of sequencers. And, you know, they can help train you on how to do it yourself, or, you know, they can run it for you. But you can also, you know, ship your DNA off to somewhere like Cornell or, you know, University of California, Berkeley, or somewhere that has their own sequencing center. And they all just have, you know, slightly different approaches, slightly different equipment. So the cost, it's very hard to kind of assess who's got the cheapest. Not all universities have their own, you know, sequencing center either. So we're lucky to have it at UMass.
That sampling.
Yeah.
So you said you need a sample to be able to do the sequencing. Does it matter, does it depend upon the organism as to where the cells are from? Or, I mean, the genetics are the same in every cell, right?
Yeah, so when we're looking at genetics, it'll be the same in every cell unless you're looking at like the germline cells, so like the ovaries or, you know, the sperm cells. Those are going to be slightly different, but you can sample genetic data from anywhere, and there are different parts of different organisms that are the best. So for like for amphibians, I've worked with salamanders a lot, and salamanders can regrow their limbs and their tails. So the least invasive way to get a sample from a salamander is to just clip off a little tiny bit of their tail. It'll regrow, you know, by the next year, and then they can go along their way. Things like mammals, so I've done some work where we were trying to get American marten and fisher cat samples, but from dead animals like that were roadkill. And we were trying to figure out, I mean, I'd never done this before. I was trying to search online for what the best piece of tissue would be to even get. And so like ears are good. So you can get like a clip of their ear, and that will help. You can even extract DNA from poop. So, you know, scat samples that animals leave out in the wild, you can take those and get DNA from them. It's hard, but and, you know, there's different quality depending on where you get something and how you store it. With genomics, you need a lot of good quality DNA. With some of the earlier approaches, it doesn't matter as much. So you just don't have to worry about where you get it from. All right. So as I mentioned in one of those answers, you know, there are these databases of genetic information that are run by the government or just, you know, organizations that are out there for the public. And you can go and you have to have a really big hard drive on your computer, but you can download genomic information from 40,000 different species, which is pretty amazing. You know, when you get down to it, there are certain groups that have been studied a lot more than others. But yeah, it's pretty amazing. And I mean, there's an even bigger database than this for really all the genetic work, so all the individual genes that you can look at. So you could get a sequence from something, like say you do get a poop sample, and you don't know what animal it came from, you could sequence it and search this database to see what organism matches up with that sample. And, you know, it'll give you a degree of certainty about that. So it's pretty neat. You know, it's a lot of data, but it's pretty cool to have. And this is run by National Institutes of Health. All right. So now I want to shift into talking a little bit about how genetics and genomics can help us solve these problems that I introduced before. And, you know, I'm just giving you a few examples. You'll hear a lot more from, you know, the next few weeks' speakers. But these are, you know, a few ways that I think are pretty cool. So one way that genetics is really useful is in helping with propagating and, you know, boosting up numbers of endangered species. You know, when a species, an entire species gets down to, you know, under 100 individuals that are left, chances are all of those individuals are very closely related. Or, you know, after many generations, they're all going to be very closely related, all their offspring. And this can cause problems if some of them have what we call a deleterious mutation. So a bad gene is one way to look at it. A bad thing in their genetic code that can actually make it hard for them to survive or to reproduce. And that just keeps compounding over time. So with genetics, we can try to do our best to figure out who should mate with who to make sure that they're going to be as healthy as possible. And this example is from the Hawaiian crow. So in between my time at Dartmouth College and my master's, I actually spent 5 months at the Maui Bird Conservation Center doing an internship. And I worked with 4 different species of native Hawaiian birds. They're not found anywhere else in the world, and there are a lot of different conservation threats to these birds. So all kinds of predators that people have brought into Hawaii. Cats are a really bad predator for birds, just house cats, but also things like feral hogs and deer and all kinds of animals that disrupt the habitat that these birds need. And the Hawaiian crow, it looks very similar to our ravens. And they're actually, they're a completely distinct species that somehow made it out to the Hawaiian Islands. And they actually were extinct in the wild up until a couple years ago. So when I was there in the mid-2000s, they hadn't released any individuals. They'd actually captured all of the remaining population in the 90s, and there were less than 20 left at the time that they did that. So they actually, all of the birds that are alive today are descendant from 9 different crows, which is pretty remarkable. And so this program it's now run by the San Diego Zoo, and they have a lot of amazing researchers that are trying to do their best to figure out, we've got these birds in captivity, who is the best match for who to have the best chance that their chicks will actually survive. And so this is a picture of 2 juveniles that were living together in an aviary. So I would feed them and take care of them. They're really, really smart, like most crows. So we had some individuals that had been in captivity a really long time, and actually when they first were brought into captivity, it was a different organization that was in charge of them, and they really kind of treated the birds like they were pets. So they would talk to them, they would let them sit on their shoulder, and that, the birds are really smart, so they started imitating human sounds and getting really attached to humans, which really wasn't good for their conservation. So now, we don't do that. Many of the descendants, they had to work really hard to make sure that they could potentially survive in the wild and have their own offspring in the wild. So this is what a chick looks like. But this is actually, it's like a little finger puppet that one of the technicians is using to feed that chick so that the chick doesn't know that it's a human feeding it. And they actually stopped doing this a few years ago and started just letting the adult chicks or the adult crows keep their eggs and raise them, from the egg. The reason they did this in the first place, though, is because they had really, low survival, so a lot of the eggs never hatched, or the chicks didn't live very long. So it was it's really hard for me being there, and I kind of left without that much hope about what was going to happen with the species. But in recent years, it's only gotten better and better. So you can see in this graph, this is the number of crows in the population, and this is the year. So 1970, there were none in the wild, none captive or released, and we only started measuring the wild crows in about 1978. So here the population just crashed But starting after that, the population in captivity just boomed. And now there are actually 11 adults that have survived out in the wild for about a year now. And this is only due to efforts to actually restore their habitat and, restore the forests that they lived in that were kind of destroyed previously.
What was the cause of the decline in the wild?
It was mostly disruption of their habitat. So the crows really like a forest that has a lot of layers. So you've got your tree canopies, but you've got smaller shrubs and a lot of different complex kind of vertical habitat for them to live in. And when things like pigs came in, feral hogs and deer and things like that weren't native, they kind of ate all of those understory plants and took away a lot of the habitat for the crows. Plus, just fragmentation and deforestation too.
What part of Maui do they live in?
So this facility was actually on Haleakala, the volcano, and the birds themselves are thought to be native to the Big Island of Hawaii. We don't really know for sure if they, how many of the islands they were on. So they don't think they actually lived on Maui, but we had our facility there, and there was another facility on the Big Island.
And so what part of, where are they in the wild on Maui?
In the wild, they're only in the wild on the Big Island of Hawaii now. Yeah.
And what about the Nene?
The Nene, so I don't have a picture, but I also worked with Nene.
Say what it is.
Yeah. So Nene are Hawaiian geese, and they are about 2/3 the size of a Canada goose and have slightly different markings. They're really cute. I mean, I really like them. And we actually had a really successful breeding program with them to where we were able to release individuals into the crater of Haleakala volcano, and they, survived there. So the Nene are doing well, fortunately.
So if you, let's say you have success with the Hawaiian crow in terms of revitalizing the population, and you put them back into the wild, if the issue is that habitat fragmentation is continuing, what's going to be the success of them once you release them?
Yeah, I mean, that's a good point, and that's why a lot of resources have gone into this conservation program and also into restoring habitat for them to live. But yeah, 100, 200 years down the line, is that habitat all still going to be there or not? It takes a lot of effort.
Yeah, in those efforts you just described, I assume this corollary benefits for other species as well in the entire ecological zone.
Yeah, of course. Yeah, so one of the big benefits of focusing really hard on one species is that, all of the other birds or other animals that are native that use that same habitat are going to benefit from those efforts. So even though the focus is more on the species than the ecosystem, we're benefiting the ecosystem.
So Hawaii has a lot of islands. And my understanding that in New England, where they have similar problems, they're trying to create some of the islands where there aren't invasive, at least animals. Is this a possibility at least for the crow in the Hawaiian Islands? Is that a consideration on the table?
Yeah, so the question is, in Hawaii, are they trying to develop pure, islands that aren't inhabited by any invasive species that can be a good kind of refuge for the crow or any other native species? And as far as I know, no one's trying to do that. Over the long term, Hawaii is a series of volcanoes, so the islands kind of rise and fall over time. So there'll be new islands eventually, but yeah, as far as I know, no one's trying to do that. All right. Well, let's move on. I want to point out this is a pedigree. So the top is kind of the oldest individuals, and then all of the lines going down, we're looking at different generations. and all the lines represent all of the inbreeding between those crows. So it's an interesting conundrum. I'm glad they're doing better. All right. So I just want to briefly touch on another way that we can use genetics in conservation. So there's this idea of wildlife forensics, like CSI, for animals, where a lot of things like ivory that are, taken illegally and lead to population declines of elephants, they'll end up being sold somewhere, and researchers can actually, take these items and use genetic tools to link them back to the actual population. And you can't see him here, but this is a researcher at Columbia University who works on this, and he says that he can actually connect the ivory to a single nature preserve where he's working. So it's pretty remarkable that you can do that, and that can help lead to efforts to prevent poaching, to just shutting down this whole process. And just another point that I wanted to make, and this is what I already sort of talked about with getting scat samples or poop samples. There are non-invasive ways we can detect species, which is really useful if a species is endangered. We don't know if it's in an area or not, and we don't want to have to catch it to see if it's in that area, because there are so few individuals. So this is just representing, all the different places on a landscape, on a glacier, in the ocean, where you could potentially collect just samples of water or soil or ice, and actually extract DNA from it and identify what species, if you have information on the species you're looking for, see if it matches up. And that's pretty remarkable too, that we can do that, and can be really useful in conservation. So now we're actually getting to my research. I'm going to probably have to speed up a little bit, but my research is really interested in fragmentation, and the flip side of that is connectivity. So how connected are different populations, just groups of plants or animals, to each other on different habitat fragments. So in looking into that, I'm really asking, how do animals move across landscapes? How does the fragmentation itself affect plant and animal species? And what can we do to prevent local species extinctions or declines? So a little bit more on fragmentation. If you just imagine that, this rectangle is showing you an area of, complete forest cover that has lots of different species living in it, and they're all happy because they can all, move around as much as they want. This would be fragmentation. So say, all of these areas were cut down and we're left with these 3 little islands or patches. So, there's a lot of different things we can assume about what's going to happen with the plants and animals that are left in these patches, but we can use genetics to actually understand what it's doing. And this diagram just shows you how we can actually create management plans that, deal with this type of fragmentation. So potentially we create a buffer around a fragment or a patch that can sort of help, help kind of insulate the species that are in there, so that they just have fewer negative effects from all the edges of those fragments. And we can also develop corridors. So, say these 2 patches were separated by just a clear cut, we can make efforts to, plant new trees and connect the 2 of them, if that's what animals need to move between them. And this is just another view of fragmentation. So just think of this as 100%, connected, and this is almost, maybe like 99% fragmented. So here, there's plenty of potential for movement. But here, the question is, what's remaining in those two little rectangles, and can they actually reach each other? Right. And this is just another, instead of a corridor, you could potentially have little bits and pieces of suitable habitat that are enough, even if they're not all completely connected, they can potentially let, animals move between the two places, even if they're not going to be actually living in those little patches. Okay. So, connectivity and fragmentation are important conservation concerns. They threaten biodiversity, and there's ways that these organisms actually, respond to the fragmentation. So we can have population sizes decreasing because there's just less area for them to live in. We can have less connections, less connectivity between the fragments, like I just mentioned. These two things together can actually lead to less genetic diversity. And genetic diversity is a good thing. The more diversity that these organisms have, the more they're potentially able to handle some kind of change in their environment that requires them to adapt. And then, when genetic diversity gets low, inbreeding goes up because there's only, limited numbers of individuals breeding with each other. So that creates, a cycle of problems for them. And really, the takeaway from my research is that everything that's surrounding that good habitat is really important too. So we're really interested in, how animals can navigate through that surrounding habitat. Okay, so I just wanted to give you a demonstration of a place that you should be familiar with. So this is Dummerston Center. You guys have a lot of really nice forest out here. I enjoyed driving through it on my way. But you can think about a species. It doesn't have to be any one species, but imagine an animal that's in site A and wants to move to site B. So it might be an animal that it doesn't matter that there's any disruption to that forest or that there's a road. If it's a large bird, for example, they're just going to fly right over that, no problem. If it's a salamander, they're going to have a little more trouble. They're going to want to stay in the forest, stay where it's wet. I know you have a number of salamander tunnels, so those are really good for helping salamanders navigate through these areas. And you could imagine a species that maybe just isn't going to deal with any of this, and it needs to stay in this really nice forest to get all the way from A to B. And that's really how I think of landscapes and try to model how animals move. And just a last bit about what genetics can do that we couldn't do without it. To track animal movement, you really have to go to a lot of trouble if you're not using genetics. And you can get a lot of really good data from this, and it's used all around the world to help conservation. But you have to have people out there catching the animals, putting bands or tags on them, catching them again. It makes it very challenging. So this is just a bird, with some identifier bands. You can see that a lot. A rabbit that has a little ear tag, for example, so you can get caught again and be identified. This is hard to see. This is a snail. So they're actually little tags that they can glue to a snail. The snail has no idea it's on the shell. It's happy. And then, when someone finds that snail again, they know it's number 87. So that's, one way you can do it. You can also use ways of monitoring their movement where you're not actually necessarily, catching them every time. But this is an example from some research that I was a technician for in Maine when I was an undergraduate, where we actually had little backpack radio transmitters on frogs, on wood frogs. And there's like a 6-inch long antenna off the back. And it doesn't bother them when they hop along. It's really lightweight. But then we would go out there with our, our little radio telemetry antenna and pick up the signal from that little transmitter. And you get closer to it and it gets quicker and louder. And so that's like a very small scale way to track movement. And we were just interested in where the frogs go after they leave the pond where they bred. So not necessarily looking for them to move to a completely different pond or anything like that. And then there's GPS collars. So these, you have to have an animal that's big enough to handle it, but this can just use GPS satellites and you can track remotely where that animal is. But you have to catch the bear. I mean, you have to catch the bear to get the genetic data anyway, but you'd have to catch the bear several times to deal with that. And then if we're talking about plants, like it just gets completely impossible. So how are you going to track a seed that goes from one tree and that a bird eats and then moves across somewhere else and gets planted and germinates? Or a single pollen grain that is on a bee. There are a few ways that people do this, but it's really tough. Genetics can kind of give us a shortcut. So it improves the feasibility and can decrease the effort, so we can spend less time actually out there chasing down animals and plants, and more time figuring out how we're going to conserve them. You can limit the disturbance to endangered populations. Again, so if you're only catching them once in their life, that's less disturbance than if you're needing to catch them repeatedly to try and understand where they're moving or whether they're still alive. And it gives us information on individuals that actually both moved and successfully bred in that population. So because genetic information is inherited, it's only inherited if you successfully reproduce. So then, an animal can move from one place to another, but then for some reason, doesn't breed, and then it doesn't contribute to the genetics of that population. And finally, we can learn about population history through genetics. Okay, so looking back at this picture, which is now harder to see, I think, and if you remember, this analogy of changing a letter in a sentence, like changing, a single base, a single letter in your DNA code, we can look at how many differences in the DNA code there are between individuals at population A and individuals at population B, and that can help us estimate how much movement there actually is between the two. So we're not actually tracking the movement path, but we can see how related they are, and then see how related B is to another population, and use the different types of habitat that's in between there to try and understand where they're moving. And this is just kind of a geometric representation of what I'm talking about. So don't worry too much about what this is, but imagine that each blue dot is a population. It's a group of plants or animals, and we would expect to see, by default, we would expect to just see straight line distance. So as the crow flies, we would expect to see them differing genetically, but there could be a lot more going on. So if we look at an environmental variable, and this is hard to see, but this is elevation. So the lighter areas are low elevation, and the darker areas are high elevation. Maybe this organism likes low elevation, so it's not going to move straight between each site. it's going to take this really long route potentially to avoid having to go through high elevation. And we can look at whether the genetic pattern looks more like these distances than it did for these distances. And that's essentially what I do with my research. Yeah, and this is just another representation of how we do that. So we can actually take a GIS layer, so we can get data from government sources and satellites that tell us things like how much forest cover is there in this area. And if I think the forest cover is really important, I can say forest cover is going to have a low cost to movement, and then open areas are going to have a really high cost to movement. And doing this, over and over on a computer, which does it much quicker than I would be able to do it myself, we can actually, walk through all the possible paths between a population, and figure out what the best one is. So the one that's going to have the least amount of cost associated with it. And different variables are going to look different. This would be, if we think our species only wants to move along streams, we would expect it to look something like this. Okay. All right, so the intro part is over, and now I'm going to talk more about my research, and I have a decent number of slides to still get through, so I may stop a little early if we don't have time, but try to go through as much as I can. All right, so the first thing I'm going to talk about is a project that I started when I was at Temple University in Philadelphia. So this is research done in southeastern Pennsylvania on grassland habitat disturbance. So the Serpentine barrens are a really unique type of grassland habitat. Have any of you ever heard of Serpentine barrens? One? Okay. So it's basically a rocky outcrop of serpentinite is what the mineral is, and it makes for really, really difficult soil for plants to live on. This is actually, these are pictures from grasslands where my species was found. And you can see it's just all rocky and dry. It has really high heavy metals, so things like magnesium, or magnesium's not one, but zinc, copper, cobalt, nickel, things that like most plants would just not be able to handle at all. But there are, some really unique species that either only live on this type of soil or can adapt to or tolerate this type of soil. And in the eastern US, there have been these really dramatic declines in serpentine barren grasslands. There are some in California that are doing a little bit better, but this area is, if you look at this inset map of the eastern seaboard, right here in the corner where Pennsylvania is meeting Maryland and Delaware and kind of near New Jersey. So there's a lot going on here. Philadelphia is up here, Baltimore is down here, and there's a bunch of agriculture out in the sort of more remote areas. So this figure shows basically in green where the potential serpentine barren grasslands could be and probably were in the past. And then these different size squares are representing how much area in acres there actually is now. And if it's an open square, it's an area where there's no serpentine barrens left. But how does this work if these soils can't really handle, or if a lot of plants can't really handle these soils? Well, it's thought that since the time that Europeans came to these areas, there have been a lot less forest fires that would actually open up potential grassland habitat. So even though we think of forest usually as good and we want to have as much forest as we can, in this case, the forest actually can encroach upon these grasslands because enough organic material will build up in the soil over time that they can actually live there. And then, like I said, there is a lot of urbanization, a lot of agriculture. Because these areas have really high metal content, there's also mining in some of them. So there's just a lot of things going against them. But there are some, really amazing organizations that are fighting to keep them, to preserve them, to keep the species there alive. So this is a little hard to see. Again, this is an aerial photo of the area I just showed you in 1985, when it was already pretty developed. You can see the darker green is forest, but all these areas along the water are more urbanized. And in 2016, you can see it's even gotten more dramatic. We don't really think it's a lot of what we're seeing is just from really recent changes, but it does just kind of keep compounding. And the species that I'm going to talk about is called Rosepink. It's this pretty pink flower that's found throughout the eastern US. It is not restricted to the Serpentine barren, so it does live in other places, but in that part of Pennsylvania, we really only find large amounts of it in those habitats. And we think that it's because they can handle the soil, so they're not competing with all the other plants that they normally compete with when they're found there. And they're interesting, they're biennial, so they live 2 years. The first year they crop up as these little rosettes, and then in the second year, the whole plant just springs out of there, and we get these nice, pretty yellow, purple, pink flowers. Wow. And my advisor at Temple University, Rachel Spigler, has been doing a lot of different types of work on this species. As I said, they're throughout the eastern U.S. or really central as well. There are occasionally these white morphs. This is more of just a point of interest than anything. We don't really know what the genetics is behind that, or what it means, but we do find that in a small proportion of the plants that we sample. These have really, really tiny seeds, so they have like a little fruit that has hundreds of seeds in it, and they all just drop down with gravity. They don't fly in the wind. They probably don't usually get kind of attached to any animals and moved. So we don't really think that the seeds are at all responsible for any genetic connectivity that we would see over any large distance. They are pollinated by what we call generalist pollinators. So that just means there's several different species of bees and flies that pollinate them. It's not a one-on-one mutualistic relationship, but they flower really late in the summer compared to a lot of the other plants in this area. So they get a lot of attention from those pollinators, that isn't kind of being paid to a lot of other plants. And this is a picture, if you can tell, of a power line actually. So because this Serpentine barrens grassland habitat really needs to be open, there are areas where it's been forested and then it's been cut down to put a power line in, and then the plants can actually do well there. So this was one of our study sites, and this is just me taking some field notes with my fanny pack there to collect tissue in. And so I collected samples from 20 different populations across that region in southeastern Pennsylvania of these species. Yeah.
Are they selfers? Do they self?
They do self sometimes, yeah. So they are able to self-fertilize. So if there aren't enough pollinators around, there's a potential for a plant to take its own pollen in to fertilize the ovule, and then it's essentially a replication of itself. That happens a small proportion of the time though, so we do see a lot of outcrossing, a lot of pollen going from different plants. All right. So I'm really curious how things like urbanization and farming, which I showed you, are affecting the connectivity between these populations. And remember, these are plants, so they're not moving anywhere, but they're relying on pollinators for connectivity. And the genetic diversity or potential inbreeding because of lack of genetic diversity within the populations. Okay, so here's just another look at the study area, and I'm just showing you this because I'm going to show you a different representation of the same area next. So you can again see a lot of spots that are cleared of forest and still a lot of spots that are pretty urban. Now, when we just transform this into different categories of land cover, we have cropland predominantly in yellow. So that's a huge amount of cropland in that area. And as we move toward the northeast, we see a lot more development. So the darker gray is kind of low-level development like housing, maybe like a farmhouse or a small neighborhood or something like that, or small business. And then the really light color represents like industrial development or kind of really urban development. And then this part is actually just the river, so we don't worry about that. And then we have dark green is forest, so you can see there's not a ton of that really left either. And if you can, there's some grass and shrubland, which is the habitat for the species, in little tiny pieces throughout the landscape. So this first area is called the Westchester Barrens, near Westchester, Pennsylvania. And this other part is state line, near the state line of Maryland. All right, so put yourself in the mindset of a pollinator. Say you go to a Rosepink plant, you get some pollen, you're trying to figure out where to go next. What type of habitat are you gonna move through? So is it forest? Forest is gonna be a place where potentially a pollinator can hang out a little bit. There might be, it might not be such a bad place for a pollinator to be, but there's not going to be a lot of those understory flowering plants because it's too dark with the canopy. Are you going to go across agriculture? A lot of agricultural crops are pollinated by native pollinators and honeybees, but in a lot of cases here we're not, they're not going there. There's pesticides. There is just a monoculture, so there's not that much for them there. Do they go to this nice farmhouse where there are some potential flowers to check out, or are they going to go through a city? And so I, again, was using this least cost path approach, as well as another approach that kind of looks more generally at the landscape instead of a single best path. I'm not going to go into any more detail on that. But just remember that, I'm using genetics and actually genomics in this case to understand how genetically different the plants are in those different populations that I showed you. So how many changes do we actually see throughout their genome? And the type of analysis I did was something that we call using SNPs. So you don't need to know this, but they're single nucleotide polymorphisms. It just means one single change in the letter of your DNA code. But I had nearly 5,000 of those from random parts of the genome, which gives me a lot of information and made it so that I didn't need as many samples of those plants to really feel good about the information I got and how it represents the population. And most of these SNPs are probably not changing genes or, changing function in any way. Most of them are just, non-coding, but are really helpful for us. So this is a bit of a different plot if you've never seen a population genetics talk before, but think of this as every single bar is an individual, and every single color is a different genetic group. So when we look at just the state line populations, we see that most of them are in this blue group. Well, there's a few that are kind of different, but for the large part, this whole area that's really well connected is really similar. But when we look at Westchester, which is more urbanized and also a little bit more spaced out for some of the populations, we see a lot of different colors there. So there's more different distinct groups, genetic groups. And that's really all I need to show you with this. And doing my least cost path analysis, this is, again, hard to see, but this purple path here is one of the best paths when I compared the genetic data to the models that I created of how pollinators might move between these populations. And this is kind of just an overview, so you can see again, we have that star always moves. We're not in New York, I promise. The star belongs down in Pennsylvania. But looking at this again, I'm going to just summarize for you what I found. So I found that urban development, whether it's low intensity or high intensity, it's always the hardest thing for pollinators to move through based on the genetic data of this plant. It's 100 to 200 times harder to move through than grassland habitat. The agriculture is slightly less of a problem, so in some cases it may not be an issue for pollinators, especially at a small distance. They can just fly over a field, but at a really big distance, this is a lot of agriculture to fly over. So it's kind of in between, might be as bad as urbanization, but might not be so bad. So what are we left with? It's really forest. Even though these plants are not found in forests, the forest seems to be the best place for the pollinators to move through. And another thing that I wanted to do was look at the populations themselves. So just zooming way in on these populations and taking into account the habitat that's immediately around them. So I was curious, I've looked at connectivity, but what about genetic diversity in these populations themselves? If they've been a little bit isolated for a while, we would expect to see less genetic diversity than in other places. And that's actually exactly what I found. So the more that we see urbanization or development and cropland combined, the less diversity that we have in these populations. So the populations that are mostly surrounded by agriculture or development are doing the worst, and the populations that have a lot of forest surrounding them are doing the best. And then for inbreeding, we expect to see the opposite pattern, and that's exactly what we see. So with more crops and more urbanization, we see more inbreeding. Okay, so to conclude this part, urban development, whether it's low or high intensity, is the greatest barrier to connectivity in this species. And that's kind of a tough thing to deal with because the crops and the cities aren't going to go away. But what can we do to best promote connectivity in this species and in the other species that may be of more conservation concern in those habitats? Crops are a barrier, especially when we look at a larger spatial scale. And forests are really the major corridors. So as I showed you before, forests, you could imagine this is a grassland, and so is this. But a corridor of forest to connect them is much better than not having it at all. And we can also see that diversity and inbreeding are related to the actual environment immediately surrounding those habitats. And I should mention that some of the most extreme ones, there's one that's actually inside a gated community. So you get like permission to drive into that community to get to that serpentine barren grassland. And I think all the people that live there have no idea, other than the main groundskeeper, have no idea what's there. So it's interesting. But others are little tiny grassland patches within a bigger forested area. So along with this, populations that don't have a lot of resources for the pollinators outside of the immediate population may be experiencing the worst declines and may experience them even worse in the future. So, potentially if we try to promote having buffers, so land that isn't going to be developed but isn't necessarily protected forest, that can actually help the species that are within that fragment. And that's a valuable management strategy that they're actually doing. So a lot of these barrens are owned by the Nature Conservancy, and they're doing a lot of work with developing buffers and actually trying to promote things like prescribed burns to reconnect some of those grasslands where forest is kind of preventing the connectivity. And what this means for the serpentine barrens themselves, this is actually a species called the serpentine aster that is only found in these habitats. It's not found anywhere else in the world. So, something, if I had unlimited time, I would love to study this species as well and see if we see the same patterns. But, at the very least, we have our data on Rosepink as a proxy for what might be happening with other species that depend on pollinators for connectivity. Okay. And now I'm just going to go through a little bit more of my previous work with salamanders. So we're switching gears. And how I moved from salamanders to plants is kind of a long story, but I really like getting to work on all different kinds of species. And there are different genetic challenges, but the theories and the conservation is all really the same. So switching from thinking about grassland, we're really thinking exclusively about the forest habitat now. Amphibians are, really the most threatened group of vertebrate animals. This is pretty dated at this point, but about a third of the known species are threatened with extinction, and 40% are declining. And there's a number of different reasons. This figure is a little complicated, but you can kind of see what the different reasons are mapping out on the map. So we'll just focus on North America. These green colors correspond to a reduction of habitat. So we see that in much of the eastern US and pretty heavily along the western US. And enigmatic declines, for a long time people didn't really know why amphibians were dying off just so much. And part of it has to do with a number of diseases that are plaguing them. So chytrid fungus is one that you hear a lot about, that's had a lot of really negative effects worldwide. And we have to be really careful, not to spread it. And now there's a new disease that's coming in and affecting salamanders. So there have been some regulations on moving salamanders across state lines and actually bringing them into the country or out of the country, just because we have to be really careful. And my study questions in this case were similar. I was curious, what are the levels of genetic diversity within populations? And I was actually looking at two different species of related salamanders. So I was curious, are there differences between the species? And then, what's good for movement? And I wasn't just looking at land cover, I looked at a bunch of other types of environmental variables that might be important. I'll show you that as well. And, you know, are the species different? So this is, you know, work from my PhD program at Washington State University with Andrew Storfer. And Washington State is, the university itself is way over next to the Idaho border, but the species that I studied were all here along the coast. This is a family of salamanders, the torrent salamanders. So there are only 4 species and it's its own family. So, you know, spotted salamanders are members of the mole salamander family, and they're similar to tiger salamanders or axolotls. Those are all part of that family. This is a family that no one really knows that much about, which is why I wanted to study it, and also why it's hard to study. So the Columbia torrent salamander is found here at between the border of Washington and Oregon in the coastal forests there. And they're actually, there's a petition to add them to the endangered species list. I've learned over time that it takes a while to get those sorts of changes made. It's been 6 years since it was proposed and they're still being evaluated. And actually, there's 2 other species I'm not going to talk about. One, the Olympic torrent salamander in the Olympic Peninsula of Washington, and then this one's in the Cascades. Those 2 are also potentially going to go on the endangered species list. The southern torrent salamander is, you know, has this much more extensive range. It's still pretty small compared to like the plant I just showed you that's more than half the country or the continental US. So, you know, they have this, you know, kind of long, elongated north-south range, and they actually, you know, right just abut each other at a single small river, and we really still don't know the exact evolutionary history of these 2 species, but I was able to find that they're really not breeding with each other. There's no hybridization, and it's probably what happened was this species probably originated in the Cascades and moved out, you know, thousands of years ago to the coast and kind of expanded out, and then there were glacial flows that actually cut off the 2 species from each other, so they're kind of separated and then they just came back together, but they're not breeding with each other. And this is the type of habitat that I would find them in. So this is a very beautiful place to work, which made it easier to talk undergraduates into coming and helping me with my research when, you know, you're just kind of wet the whole time and sometimes frustrated. So this is actually kind of a more open area, but this is a stream where I found salamanders. These guys have really limited dispersal, so limited movement abilities. They, you know, no one had really studied their genetics on this level before I looked at it, but you know, some of the ecological data suggests they're only moving, you know, at most like 100 feet or 100 meters actually, you know, in their regular home range up and down a stream. And we really didn't know how far they're going to move to get to a different stream at, you know, when they want to go reproduce. And they have really specific habitat, so they breed in the streams. You know, there are a lot of salamanders that do that. These guys actually have a larval stage, so it's like a tadpole stage, where they stay in the streams for about 3 to 4 years after they're born, which seems like a long time. I mean, these are really nice, pristine headwater streams, so I guess there's just not a lot to disturb them other than larger salamanders that might eat them. And then they eventually metamorphose just like a frog would, and then can move across land, but they're usually still found near the streams as adults. And they're really associated with old growth forest. So they like it cool and wet. This, you don't need to pay too much attention to what all of this is, but these are just a series of different types of habitat variables and environmental variables that I tested. Canopy cover is another one, again, that's kind of easy to think about. Some of this, like precipitation here, is basically precipitation that falls as rainfall and not as snow. You can see there's kind of a gradient in this example, but it doesn't vary that much in these population or in these species ranges. I looked at a number of things that are related to elevation, because we don't think that a little salamander, they're really, as adults, are only about this long. So they're much smaller than a spotted salamander. We don't think that they would really want to go up a mountain and down the other side to get to the next stream. So we looked at a lot of things with elevation, and then road cover and land cover, and stream cover. So heat load index, it's kind of a measure of how much solar radiation is going to be hitting a surface. So if you think about the angle so whether something's facing north or south, if it's facing north in the northern hemisphere, it's going to get less direct sunlight than if it were facing south. And this also takes into account the angle, the slope, and I think one other thing. It's kind of a combination of things. Okay. So this is what my sampling actually looked like. And this is, a pretty huge area from Washington down to Northern California. These samples are not at the very southern end of their range, but this is kind of an area with some national forests that's really good habitat for them. So the blue is all the southern torrent salamander, and the yellow is all the Columbia torrent salamander. Ideally, I would have samples from this whole area, but I spent months looking for good streams and good habitat, and previously identified locations for them in a lot of these areas that are now clear-cut or developed in some way. So the major industry here is timber harvest. There's a lot of the areas I worked in are national forest, where they're thinned in some cases, but then other parts are privately owned, and there's kind of this whole mishmash. And there's sort of different regulations in the different states, so it gets complicated. All right, and I used a different type of marker this time, a different type of genetic tool. So instead of looking at across the whole genome, this is more of an older genetic approach called microsatellites, and you can ask me about that later if you want. But I only use 8 to 10, and each one gives us a lot more information than a single SNP that I showed you before, but it's still limited. So I had to have a lot more samples at each population, and there's still things that were kind of hard to tell from this. So that's why moving toward genomics is really useful. All right, and this is my summary slide here. This is just showing the sampling again. And I also got some samples from collaborators at the Forest Service in Oregon, which kind of helped flesh out my sampling. And it's nice to have that person to actually talk to about the management of what's going to be done with this data. And these are all pictures from sites where I worked. Basically, when it came down to it, these are the 3 types of habitat that most explain genetic distance or genetic differences between populations. So it seemed like open areas were bad. This is actually a spot where there's a stream right in there, and there's a buffer remaining around the stream. There are a bunch of happy salamanders in there. That was one spot where it was easy to actually catch them. But the whole area around there is clear cut, so it'll be interesting to see how that population does over time. Is it connected enough with just those stream corridors? And this is a picture of a road. This is a road in the middle of really dense, nice forest, so that road probably isn't a huge problem for salamanders to move, but bigger roads, again, you guys are familiar with salamander tunnels. In some cases I have, highways or, smaller highways at least in between populations. So there are still things like culverts, but if you're a little salamander and you're on a really steep slope in your stream, you're gonna go down the culvert much more easily than you're gonna come up. So there's interesting dynamics with that. And it's not a surprise that having stream habitat is a good thing for movement. So our data really reflect that they actually are moving alongside the streams once they metamorphose, and they're not moving like across a mountaintop or on a really rainy night just going really far. So that was interesting. It's still kind of hard to picture, but yeah. And I also found that this is just an example of one of my more southern sites in the southern torrent salamander range, where there's pretty contiguous habitat there. And then this is one of the more disturbed sites in the Columbia torrent salamander range, where we do see somewhat lower genetic diversity. It's still kind of we need to gather more information. Ideally, use some more genetic tools to really figure it out, but that's the pattern that I overall saw. So, there really weren't a lot of differences between the species, but they both do seem to have pretty limited ability to move and limited connectivity. And this, I should point out, we can tell based on different measures that we can compare to other species. So those values of connectivity are pretty low in this species. They like to minimize dry areas and elevation change that might inhibit their movement. And really, it seems like stream buffers are one of the main ways to improve their connectivity. So if they're really moving along streams and land is being used for timber harvest, maybe that doesn't matter that much to them as long as they have a nice buffer, keeping their stream cool and happy. Yeah.
Sarah, do you have anything to say about the width of the buffer?
Yeah, so that's not something I studied, but it is in all those different areas, like what's actually required varies a lot. And I would think that ideally maybe like 100 meters of buffer, that would probably be a lot. At least like 30 meters of buffer. But yeah, in some places the rules are like as little as 8 meters, I think. Yeah. Oh.
I was wondering if we're at the point we should open up to general questions and conclusions, given the clock.
Sure. Yeah, let me just show my final slide here. This is all about the work that I'm kind of working on now and in the future, so I can talk about that with anyone individually. But I want to leave you on a happy note, which is that people are thinking about connectivity on a really broad scale now, and doing things like building these huge bridges over highways for wildlife who move big distances is one of the ways that we can try to maintain that even as we continue to use the land for our own uses. So this is just showing, the darker colors are showing the more important corridors for wildlife movement that, really like trying to protect those areas would protect the movement of the most number of species.
I do have one specific question.
Mm-hmm.
While we're digesting the acknowledgements.
Yeah.
And that is your work here at UMass. What sorts of questions are you looking at that you might come back a year from now and tell us you have some answers?
Well, I do have slides for that. So I have two different projects I'm working on. I don't have results to show from either of them, but this is work I'm doing with my advisor, Chris Sutherland, who's in the Department of Environmental Conservation. He is from Scotland. He did his PhD research on the water vole in Scotland, which is a beloved conservation icon in that country. Their water voles are found in Europe as well, mainly in Europe. But there are these, they live in these little population networks, and this is an example. This is showing all the different populations that have been sampled by our collaborators. But they don't actually inhabit all these populations every single year. So there's a ton of movement between them, and sometimes they blink out, and then a few years later they blink back on. So it's really interesting, and it's really hard to study because they move so much. It's like the opposite problem that I've had before. And that's why we're going to get a lot of data and actually try to reconstruct their pedigree to say, if an individual from one year is related to an individual from the next year in a different spot, that gives us an idea of who moved where. And the other project that I am working on collaborating with Lisa Komorowski is on alewife in the St. Croix River in Maine. So there are a number of dams that have been built, and there are actually some landlocked populations above those dams, but they're normally a fish that, like salmon, will be spawned up in the streams and then go down into the ocean to develop over time and then come back up the streams as adults. So it seems like there's some that are just trapped upstream, but now there's efforts being made to fix the fish ladders that actually allows the alewife to move up. And we're really curious what's going on there. Are there really two different separate populations? Or did the landlocked ones come in from somewhere completely different, which is something that happened in the Connecticut populations of this species? So it's really interesting. We have a bunch of samples from the different dams from about 15 years ago and recent samples that we're going to compare.
Well, it's getting close to our normal closing time, and we want to be sure to get you to lunch on time for your afternoon appointment at the Dummerston School. But I do have one final question, or actually a comment that you might make about our two speakers who are coming a week from today. Mm-hmm. Lisa Komorowski, whose name you just mentioned, and Nadia Fernandez. And since they're your colleagues, who better to introduce a little bit about them than you?
Sure. Well, yeah. So Lisa is a relatively new faculty member at UMass in environmental conservation. And she's sort of taken me under her wing because she's the one person in the department that does genetics and genomics. I actually started working in her lab some when I was helping a student with a different project. But yeah, it's been really great. She's, she does all kinds of amazing conservation research on fish and sea turtles and other species. So it'll be interesting to hear from her. And then Nadia is one of Lisa's PhD students. So Nadia has a lot of expertise with genomics looking at golden eagles, and I think that's what she's going to be talking about. So she has a lot to teach me about having lots of data that I never had in any of my samples, but I think it'll be really fun to hear from both of them.
Is there an urgent parting question from anybody in the audience?
Something there.
Well, I don't know how urgent it is, but going back to the salamanders, one of the things that you stimulated me to think about is the salamanders don't do the 100-yard dash across, miles and miles. It would seem to me that the headwaters are the areas where if they're in that area, where they can easily go over to the next headwater to move down in that area and populate that. And it seems to me that some of your genetic methodology would be able to explore these critical areas of conductivity and movement of species like the salamander. Maybe that's just way off base, but would
Well-
seem to make some sense.
Yeah, so that's a good point, and really what the data showed us was comparing just a straight line from one population to the next. Granted, those are not always, neighboring streams. Some of them were much farther apart from each other, but that explained less of the genetic data than a path where they actually moved all the way down along the stream network to a larger river and then up another headwater stream.